Welcome to Measuring Carbon in Non-peat Soils. This video demonstrates field sampling for non-peat ecosystems and describes how to properly package the soil samples to be transported and analyzed in a lab. Of the carbon found in terrestrial ecosystems, the majority is found in soils. In Canada, non-peat soils are estimated to hold over 208 billion tons of carbon within the top 1 meter. To estimate these carbon stocks, let's dig into the different methods for measuring soil carbon. In this video, you'll learn about site selection and preparation, the 3 methods for sampling the carbon stored in soils, and the steps for processing and analyzing samples in a lab. The 2 most common methods for measuring carbon in soils are the soil coring method and the soil pit method. Both methods achieve similar goals, allowing you to sample soil at different depths and analyze those samples for carbon content. The third method, which we'll also cover briefly in this video, describes how to take samples from very shallow soil. For each method, there are 3 steps to collecting accurate measures of the soil carbon stock. First, separate the soil into distinct layers. Second, obtain the bulk density of samples from each layer. Finally, determine the carbon content of the samples. This video will cover the basic elements of each step, but you can refer to the accompanying guide for more information. Before digging in, we recommend that teams have at least 3 members to ensure the process is efficient and safe. While this video does not cover the later analysis of soil sample data in the lab, it's important to plan for lab analysis in advance to ensure the data you collect meets the end goals of your project. The first step when preparing is to think about site selection and sampling logistics. Consider the following questions. Where is the study area located? How many sites are needed within the study area? How many samples should be collected at each site? How deep should the soil cores be? A soil probe can be a useful tool to survey the site before selecting a sampling location and may inform the sampling depth. For detailed methods of site selection and sampling design, please see the accompanying guide. Once you've selected your site, you will need the following materials to obtain soil samples using the methods described in this video. Now you're ready to head into the field. Once you've arrived at your site, you must document the vegetation using the following standardized photo series protocol. Start by documenting vegetation and canopy cover by taking 2 photos of the site, one pointing straight down and one pointing straight up. From the same spot, turn to each cardinal direction and take 3 photos, one parallel with the ground, one angled 45 degrees up, and one angled 45 degrees down. Once finished, you will have 14 photos of your site, 3 photos for each cardinal direction, one documenting vegetation, and one documenting canopy cover. Be sure to record the site where you are sampling with a waypoint in your GPS and record the coordinates in your notebook. Regardless of the number of samples you are taking, we recommend a systematic approach to recording your core IDs. You can come up with your own, but we recommend using a method that identifies the location. In our example, we use PE to note Peelwanuck, then the site, in this case it's our first site 01, and then the sample. In our case, it's the second sample, B, from our first site 01 in Peelwanuck. If a surface core is available, we recommend using the following coring methods to obtain soil samples because it is more effective for collecting the entire core. Before coring, find a flat area close to the coring spot, lay down a tarp, and prepare all the required equipment. Once you have selected a coring site, gently push the core into the soil, keeping it as straight as possible. Corers with serrated edges can be used to cut through coring spots with tough roots. With the corer fully inserted, twist and jiggle the corer to release the bottom part of the core sample from the base sediment. If the core gets stuck, dig down from the outside of the core to give the corer more room, or cut through the ground with a trowel or knife. When lifting the corer out of the ground, Try to keep it as straight as possible until it is fully extracted. You can now transport the soil core to the processing area. The core is now ready to be revealed, measured, and photographed. Push the core sample from the bottom using the core extraction tool so that the core is revealed through the top of the corer and is caught by the sleeve in the process. Place labeled end caps on the top and bottom of the core. Secure the caps with tape. Measure the length of the core and the depth of the hole from which your sample was taken. Make notes of any distinct changes in the soil profile in the core. Record this data in your notebook. Label the core sample and place it in the cooler. After fieldwork is complete, all samples should be transferred and stored in a freezer as soon as possible for lab analysis. The second method for obtaining soil carbon measurements is to dig a soil pit and collect samples from each soil layer individually. Here's what you will need. At the selected site, use a shovel to dig a hole in the ground that is at least 0.5 meters wide. The hole should be deep enough to expose all the soil layers and should be wide enough to easily collect samples from each layer. You know you have dug deep enough when you reach clay or if you can't dig any further. In your notebook, record details for each soil layer, including its depth interval, color, and texture. This will be used for soil classification. Then, using a small soil sampling ring or bulk density disc with a known volume, collect a sample in the middle of each soil layer and place it in a resealable bag. This will be used to determine the sample's bulk density and carbon content. The last method for obtaining soil carbon measurements applies only to very shallow soils, such as those found throughout the Canadian Shield. While this method is less precise than coring or digging soil pits, it can be useful in the right conditions. Here's what you will need. This method uses a medium-sized frame to dig a small pit, typically 10 to 25 centimeters long. Additional details of the frame can be found in the accompanying guide. From here, collect the entire soil sample all together and transfer it to a labeled resealable bag. Once the sample is collected, fill the hole with material of known volume, such as water. The volume of water replaced in the hole is the soil sample's volume. Record the sample number and volume in your notebook. Package, label, and store your sample in the cooler until it can be transferred to a freezer for lab analysis. In the lab, samples will be weighed, dried, ground up, and analyzed for physical and chemical properties. A detailed procedure of the lab protocols can be found in the accompanying guide. Once samples are analyzed in the lab, you'll be able to assign a measure of the amount of carbon that is stored in non-peat soils for each site. See the accompanying guide for details. Congratulations! You now know how to take a non-peat soil sample. In this video, you reviewed the steps for selecting a study area, site, and plots, preparing your plot for coring or alternative sampling methods, taking a soil core or other soil sample types, and processing and packaging your soil samples for lab analysis. For more information, please consult the guidelines for non-peat soils and refer back to this video anytime. Happy sampling!